We're all a bit afraid of it. Soon it will become society's biggest challenge. So can science help us to deal with the problem of growing old?
Time takes its toll. This is how most of us will end up, the steady decline of our bodies into ill health and frailty. Cells cease to regenerate, and one by one the organs decline as the body's normal operating system shuts down. But what if there were to be a form of therapy which could turn back the clock? Clive Svensson, a cell biologist, may have made a breakthrough that could create a medical revolution.
Well, I don't know. Be looking at some cells. How big do you think a cell is?
That's very big. You look through the microscope.
Yeah, it's very small. We'll be looking at brain cells. And the brain cells will be growing in the test tube.
Dr. Svensson is working with some of the most fundamental cells in our body. They're called stem cells. Life starts with stem cells. They appear in the very first days after fertilization and are the essential building blocks for our bodies. Just these 50 to 100 stem cells have the ability to grow and divide until eventually they'll give rise to every organ, tissue and cell in our body.
During development of the body, there has to be a cell which organizes itself into the tissue. So, for instance, with a kidney, there has to be a starting cell for the kidney, and that cell has to give rise to the millions of cells that make up that kidney. And so they're very interesting in terms of medicine because we know, looking at stem cell biology, how organs actually came into being, and perhaps if we can understand that process, if that organ goes wrong, we can repair it using stem cell biology or using stem cells, perhaps transplant it back.
In theory, any degenerative disease could be treated with stem cell therapy. John Ardley has an incurable illness of the brain and one of the most terrifying of all, Parkinson's disease.
First, two things really we noticed first of all. One was that I was not walking very well, and I wasn't swinging my arms like people do when they walk. My wife noticed that as well. And the second thing was that my handwriting deteriorated to the point when I went into a shop to pay for something on a credit card, and the shop wouldn't accept my signature. So my wife told me to go to the doctors and find out what was wrong. And after some clinical tests, they diagnosed Parkinson's disease as the problem.
So in Parkinson's disease, we know very clearly the problem. The problem is that these patients have lost a specific type of cell in the brain called a dopamine neuron. And one idea to treat this disease has been to replace those dopamine neurons with dopamine neurons from post-mortem fetal tissue, from elective abortions. And the fetal tissue contains dopamine neurons at the right stage of development, that if you transplant them back into the patient, they start to make dopamine neurons.
Trial operations have shown the technique works. Fetal stem cells were inserted into the brains of Parkinson's sufferers, where they soon started to develop on their own. Follow-up brain scans showed that the stem cells had survived and were producing the missing dopamine, thought to be a key to preventing the onset of Parkinson's. But the use of human fetuses for medical treatment is highly controversial. Each transplant operation needs tissue from as many as 8 fetuses. So, despite their awesome medical potential, harvesting stem cells from discarded human embryos could prove to be a step too far. The prospect of reversing many aging-related diseases may never be realized unless another way of growing stem cells can be found.
Unfortunately, it isn't as easy as that. Stem cells have proved very difficult to grow. But then Clive Svensson had one of those inspired scientific moments.
Well, we were struggling to try and grow these cells in the culture dish. And actually, I was at home in my kitchen one day, and we were making some carrots and chopping the carrots. And I was thinking, how can we make these spheres of cells smaller without breaking them to single cells? And just the idea came from chopping vegetables well, why don't we try chopping these lumps of tissue? So we take what's called a tissue chopper, which has a razor blade on it. And the tissue chopper then does this automatically, rather like a kitchen slicer and dicer, slices the tissue into cubes, and those cubes then will go on expanding for weeks, months and years. I tried it as just a technique to see if it would work. I was not really expecting any great results. And then when we saw that every time we chopped something into 4, each one of them came back to the same size as the mother sphere after 2 weeks, we got quite excited and thought, this is great. And we found using this technique that cells remain in touch with each other. And cells love being together, they hate being on their own. So when they're together, they seem to grow very happily. And this relatively simple technique actually allows you to grow enormous numbers of these cells. And they keep the ability to make neurons for a long period of time. So we've managed to get 1.5 million times expansion. So from one fetus, we can go to 1.5 million within about 150 days.
For people like John, Clive's breakthrough offers real hope of a practical cure for degenerative diseases.
So we're trying a new approach, which is to actually obtain stem cells in culture, which are the precursor cells for the brain. And we take them out from the developing brain and trick them into continually growing. And those cells then, you can see, we can actually build up stocks in the test tube. And those stocks of cells can be then used in transplants. Now, we're at the point at the moment where we can actually generate lots and lots of cells, human cells in culture. And we're currently trying to turn them into functional neurons.
How long before people would actually start to benefit from this research, do you think?
We could discover the way to make dopamine neurons next week, or it could take 2 or 3 years. And it's very difficult to say from then. You have to go through a series of functional tests in animals, and finally into the patients if all the tests work.
Even someone of my age, 54, maybe 60 or 70 before this is widely available, of course. This is the...
Well, even if it's 10 years, I mean, then you might benefit from it at that stage.
With important ethical questions resolved, the path will be cleared for stem cell treatments to extend and improve the quality of our lives. The next generation will still get old, but they will stay healthier for longer.
We're on the sort of cutting edge now, the new millennium, that we're just beginning to understand how we come together as cells. And this sort of technology, I think, will be driving research in this area over the next 20 years. And cell therapy will be at the forefront of what comes out of it.
Unfortunately, racing in the 50s was very dangerous. The Argentine Grand Prix, tyre wear was a big problem, and I started to wear the rubber away to the canvas. I was absolutely flat out at 140 miles an hour with tyres in shreds, but it isn't that easy when your life's at stake. I was going onto the grass to save the tyres, and I tell you, that was really the most exciting but frightening race I've ever done.
One of the hazards of aging we all face is the increasing likelihood of strokes, distressing episodes that may leave the sufferer with long-term paralysis. But help may be at hand in the shape of a new way of stimulating paralysed muscles.
Rob is recovering from a stroke. He has lost movement in his legs and in one arm and is undergoing painstaking therapy to improve his condition.
That's it, good. Okay, yeah, remove it completely. Good for you. Stand tall and step forward. That's good. Good, Rob.
However, a remarkable new treatment being developed in North America could give him a new lease of life. In charge of the project is Dr. Gerald Loeb, Professor of Biomedical Engineering at the University of Southern California. Strokes are the leading cause of disability among adults, yet therapy to aid recovery is expensive and often difficult for the patient. Damage to the brain can inhibit motor abilities. Unused, the muscles in the arms and legs will quickly shrink and wither, leading to partial or full paralysis. There's also the danger that with weakened muscles, limbs will dislocate, so patients need constant physiotherapy. Electrical stimulation is also used, The muscles are twitched and therefore exercised further by controlled bursts of electricity.
And you'll feel a tingling, pins and needles sensation under...
All of which means patients must spend many of their waking hours in hospitals and clinics, receiving time-consuming and often painful treatments.
It'll go off for 5 seconds, and it'll just cycle on and off like that. So it's on now.
At Queen's University in Ontario, Canada, research is being carried out into an exciting new device that could be a quick and effective alternative. The first clinical trials are underway. And the focus of all this work? The Bion. It's an electrode not much bigger than a grain of rice.
In a stroke, the brain loses the ability to control the contractions of muscles. The muscles are still present, they're still innervated by motor neurons from the spinal cord, but the brain can't turn them on. And so the Bion is a way to restore the ability to contract muscles by replacing the missing electrical signals at the level of the motor neurons.
Inserted deep into the muscle tissue, the tiny Bion will, in effect, provide the electrical stimulation that should come from a healthy brain. Today, Rob will become only the second patient to undergo Bion therapy. Dr. Stephen Bagg, director of the Stroke Rehab Unit, will lead the surgical team.
He's the typical person who is at risk of developing a partial dislocation or subluxation of the shoulder because he does not have that muscular support to prevent the ball from gradually pulling down and partially out of the socket. Okay, here we are.
In a normal shoulder, the arm fits snugly in the socket, but in the shoulder of a stroke patient, the arm can hang painfully low as the muscles weaken.
The benefits that I see of this technology over what previous studies have shown, the previous studies use surface stimulators, electrodes that stick on the skin or percutaneous wires that go through the skin and into the muscle. And that technology requires a lot greater stimulation, electrical stimulation, in order to cause the muscle to contract than with the Bion. So it's a lot more painful, a lot more inconvenient, I think. Looks like we're ready. Okay, so again, Rob, after freezing the skin in the insertion site, we make a small incision and then insert the needle, and that needle has to be large in order to let the Bion slip down through the hub of the needle. Just a very slight, can you take it up to 3, please? The reason we stimulate is we want to make sure that that needle, that the Bion is placed in the motor point or close to the motor point of the muscle. And the motor point is just that area in the muscle where there's the highest density of nerve endings. So you're going to get a bigger muscle contraction for the amount of current that's there. Once we're comfortable that we're in the motor or close to the motor point, and we pull out the guide for the needle, leave the catheter there, and then introduce the Bion, and then push the Bion down to the end of the needle, pull the catheter out, and the Bion remains in the muscle. That's it, Rob.
That's good.
Do you see us twitch? No. Okay. His elbow's twitching.
Good. So it looks like it's working, Rob.
Mm-hmm.
So we just let the tissues heal for the next 5 to 7 days, and then we get going.
Okay.
Sound good?
Sounds good.
Okay.
After the successful completion of Rob's surgery, Dr. Loeb visits the home of his first patient, Richard, who received Bion implants 8 months ago. Since his surgery, Richard has continued the Bion therapy unassisted using a device called the personal trainer. It's a microcomputer. At the push of a button, a signal is sent from the coil he wears on his shoulder to the Bion. It means the patient can stimulate and exercise his weakened muscles while at home.
It's not like having a massage or something like that, to be quite honest. There's no real sort of pain in it at all. And afterwards it feels as though, you can move a little easier.
In an X-ray of his arm, the Bions can clearly be seen. After just 6 weeks of therapy, the muscles have improved and the arm has moved back towards the socket.
He's certainly been very helpful in helping us to understand how the treatment feels, how he feels about it, how to improve it, what sorts of treatment regimes patients are going to find useful and effective.
Well, it feels a great privilege in a roundabout way to have this opportunity to help other people beside myself.
It's still early days for the Bion, but ultimately they could have many uses. From replacing heart pacemakers to reversing full paralysis, they look set to become a valuable tool in the battle against aging.
Gradually, science is helping us stay healthy and active for longer. But will we be satisfied with this, or will we look to science to make us immortal?
This woman knows how to look after herself. She's fit, healthy and looking good. But would you believe she was over 100 years old? Fiction today, but it could be tomorrow's reality. Most of us would like to be fit, healthy and to live longer, but at the moment, to be healthy in our old age often requires a lot of hard work.
I enjoy I love doing exercise. It feels good. I feel that I'm doing something that's good for me.
Well, I worked out all my life. I mean, I'm just used to doing it. It's part of my life.
I'm 81 years old, and I come about 4 or 5 times a week. And when I don't come, when I'm on holiday for a couple weeks, I really notice the difference climbing the stairs.
But all this effort may soon be unnecessary. While living forever may never be feasible, it may soon be possible to extend our lifespan by decades and have a better, healthier standard of life without working up a sweat. Researchers at the University of California in San Francisco have recently made an astonishing discovery that the rate at which animals age can be changed. They've studied a small nematode worm, C. elegans, and have managed to dramatically lengthen its lifespan by altering just one single gene, a gene called DAF2, whose job is to make a protein which acts as a hormone receptor. Dr. Cynthia Kenyon explains.
The first thing that's interesting about the fact that the DAF2 protein is a hormone receptor is it says that aging is caused by hormones, for sure, at least in this little worm. Because what we've done is we've changed the receptor in the laboratory so that it doesn't bind to the hormone as well. And as a consequence, the animal has a lifespan that's twice as long and stays young much longer than normal. Now we enter the worm nursing home. This worm is a 13-day-old adult, so this is just about a 2-week-old worm. And you can see it looks old. That's the first thing that you see. It's not really moving, only its head moves just a little bit. Its body is kind of pockmarked. Now you see the mutant worm. In this worm, we've changed the DAF2 gene. So this worm is going to live twice as long as normal. But this guy looks much more youthful, more vigorous. It's moving. It seems interested in the world. If we could make a change like this in humans, then it would be as if a person who was 90 years old looked and behaved and felt like a 40-year-old.
But how does this work? How does the DAF2 hormone receptor regulate aging?
In our laboratory, we found that there are signals from 2 different parts of the animal, the little worm, that regulate its lifespan. The first kind of signal is one that we think is controlled by sensory perception.
Let's look at the worms in human terms. Normally, worms are able to smell and taste food in their natural environment. But when they are genetically manipulated so they can't smell or taste as well, their lifespan increases by 50%. So, in human terms, they'd live to be 120, but not be able to enjoy tea time.
We've also found that signals from the reproductive system affect the lifespan of this worm.
When the worms were manipulated so they couldn't produce sperm or eggs, their lifespan increased by 60%. In human terms, they'd live to be 150, but any interest in the opposite sex would be a waste of time.
So not only did they live longer, but they stayed healthy and young longer. So this had a youthfulness effect on the worm. So the reproductive system and the sensory perception system, the nose, both of which affect lifespan, and both of which appear to affect lifespan at least partly by affecting this level of hormones that interact with this DAF2 receptor. So we think that this receptor is playing a very central role in determining how fast the animal ages.
Well, that's good news if you're a worm, but could this ever happen in humans? Do we even have a human version of the DAF2 gene?
The answer is yes, but manipulating this single gene-regulated hormone receptor on its own is not going to allow us to cheat death. For the complete picture, we also need to look at aging in animals more closely related to us.
A team of scientists in New York have made a breakthrough discovery. By mutating another gene called P66, they've extended the lives of mice. Dr. Pier Paolo Pandolfi heads the research team.
So a mouse on average lives 2.5 years, if you're lucky 3, and our mutants would live on average 30% longer, which doesn't seem that much if you translate this into the life of a mouse, but is a substantial achievement if you calculate what this might be in a human being, which is approximately 30-40 years longer.
So, how does the P66 gene affect aging in mice? Normally, P66 produces a suicide enzyme, which makes a cell self-destruct when it's been damaged by stresses in the environment. When the P66 gene is switched off, the cells don't die, and the mouse's organs are better protected from environmental stresses.
It is intuitive that if you, throughout life, you're exposed continuously to stresses, these cells would be damaged throughout life by these stresses, and this might result in the generation of specific organs, leading to reduced lifespan. It is therefore intuitive that if you can cope with these stresses better, you might live longer.
So it looks as if by manipulating genes, we can hope to slow the aging process. But does this mean gene therapy will keep you looking good and feeling fine forever? Perhaps not yet. Aging is the result of the interaction of many different factors. But Dr. Pandolfi believes his team's discovery may soon lead to effective treatments for some of the most common age-related diseases.
A possible implication of our finding is that the understanding of this genetic mechanism might give us a hint in developing therapeutic strategies which would antagonize these degenerative disorders.
So, although we may never live forever, we can look forward to a greatly extended and much healthier lifespan. Being 50 and feeling 20 may not be so far away after all.